Hey, thank you very much, Emmanuel, for that very gracious introduction. It's a great pleasure to speak with you all. And I would obviously much rather be there. But I also appreciate that you might at 4 o'clock on a Friday afternoon want to be elsewhere. So I appreciate the attendance. So thank you very much for coming. So what I want to do today is begin with the point that I think that ecology is really um, at an important place in its development and really at a place that's quite good. And it's in a place that's quite good because the different motivations that we have for doing this kind of science have really converged to give added interest and importance to the science that many ecologists are doing. So on the one hand, we have an ecology that's been motivated by a fundamental curiosity about how nature operates. And this is sort of typified by the explorations of the great naturalists of the 1800s, but I think also extending well through the tradition of MacArthur and others, where we're really motivated by, we want to understand how diversity is structured in nature, and we're just inherently curious about that diversity. But we also have ecology that's motivated by the need to solve global environmental challenges. So problems such as climate change, the spread of invasive species, and the emergence of infectious diseases have play increasingly prominent roles in mainstream ecological journals, but they also I think are playing disproportionate roles in attracting sort of the next generation of ecologists to our field. If we look at the students that we're recruiting to our labs, these types of issues are driving the directions of their theses. And what I want to do today is argue that progress towards solving these global environmental challenges with ecology is going to require continued development of the kind of science that has been traditionally motivated by this fundamental curiosity about how nature operates. And I'm going to try and make this case by giving some examples from our own work that have been motivated by the question of how plant communities will respond to climate change, this sort of lower issue down here. So widespread recognition that changing temperatures and changing patterns of precipitation are ongoing and are going to magnify over the current century have motivated many ecologists to go into nature and try to conduct experiments like the one you see here, where we try to mimic a future climate condition here, a drier climate, and we try to evaluate how populations, communities, and ecosystems will respond to these changes. Now, as a community ecologist, someone who studies the interactions between species, one of the most exciting developments that has emerged from this body of work is that we try and understand how these climate changes affect individual species. We find that while there are certainly sort of direct effects of climate on the fates of these focal species, very often some of the most dramatic impacts of climate are indirect through changes in the competitive environment these plants are facing. And one of the messages that I hope you leave with today is that these indirect interactions of climate change can be important, but more significantly, what we think we know about these indirect effects of climate change is really just the tip of the iceberg. And it's the tip of the iceberg because what we've done thus far is study the changing interactions that occur between plants that are currently found together, that are found together underneath one of these rainout shelters for very obvious practical reasons. But when we think about the types of competitive changes that are going to be most dramatic in shaping the fates of these species under climate change, it's probably not going to be changing interactions with one's current competitors, but instead it will be the changing interactions that are going to emerge when entirely novel competitors migrate into this habitat as it becomes newly suitable for different sets of species. So let me explain to you what I mean by this in more detail. So here's a diagram of different plant species situated along an environmental gradient, where each one of these histograms is meant to correspond to the range of environmental conditions that each one of these species tolerates. And here's the gradient today, in what we call the present. And it might be an alpine gradient underlain by climate variables such as temperature or rainfall. But we can expect that if the climate changes in a way such that locations that are experiencing this environment today are now experiencing that environment over here in the future, we expect taxa to migrate in a general upslope direction. But the key is that this migration is not going to be synchronous. So we might expect, for example, that some species, such as the red one, will migrate very effectively, and others, such as the blue one, will migrate very poorly. And this, of course, comes down to differences between the species and their dispersal capacities, as well as their demographic potential. But what you end up with in the future then is an interaction between two species, the red and the blue, which don't interact in today's environment. And this is what we call novel competitive interactions for the purposes of this talk. Now, I think a fair bit of ecological natural history would lead you to believe that these novel competitive interactions could play decisive roles in shaping species' fates under climate change. But we actually have very poor empirical validation of this principle. So what I want to do today is build a talk around these novel competitive interactions. And what I'm first going to do is ask sort of the most obvious question, which is how do these novel competitors shape individual species' responses to climate change? Does, for example, the arrival of this red species lead to the loss of the blue in our simple cartoon as I'm showing you here? But next, what I want to do is come back to the processes that are actually generating these novel competitive interactions, and that's the asynchronous migration. And I want to ask how well we can actually forecast the migration of taxa under climate change. And in particular, I want to ask, can we predict species migration velocities with currently measured vital rates, currently measured dispersal and fecundities, for example? Will this provide the information we need to forecast where species will be in the future and which taxa will actually come together in the future, generating these novel interactions? And then last, I want to ask, if we can forecast which of these taxa will face one another, let's say in 100 years' time, can we predict the outcome of that interaction with knowledge of the plant's functional traits, their height, their leaf traits, and so on? Okay, so let me begin with this novel, this first question here concerning how novel competitors shape species responses to climate change. And as I've indicated already, we don't have very good empirical validation of the idea that these novel competitors will play decisive roles, even though our natural history intuition might lead us to suspect that they would. And there's basically two problems with there's two challenges that get in the way of empirical validation. The first is that we tend to have poor predictions for which novel interactions will emerge in the future. So if, for example, I'm standing in a meadow in Montpellier, and I want to ask, what will the plants that are at my feet today face in 100 years' time, it's not entirely clear what those plants will actually be. Will the plants be able to migrate the distances required, even if we have a good expectation of what plants might enjoy Montpellier's climate in the future? The dispersal question becomes quite challenging. And then even if we can predict which species will be interacting in the future, we need to be able to create those interactions today and do so under the climate of the future. And this creates some experimental tractability challenges. So again, these are sort of two major reasons why I don't think we have good empirical validation of the idea that these novel competitors might play decisive roles in shaping species responses to climate change. So I think that we can partly overcome these hurdles by working in alpine environments. And that was certainly the idea that we had, myself, Jake Alexander, and Jeff Dietz, who are now professors at ETH Zurich and the University of Oregon, respectively. So we were working in the eastern Swiss Alps. Here we have our field site near Chur. And what you can see is our field site at 2,000 meters, just above tree line. And you have a diversity of very nice alpine plants, many of which overlap with what you would see in the French Alps. And these plants face a climate in 100 years' time that is comparable to what they face today if they're able to migrate 600 meters upslope, so near the top of this ridge here. And it's not unreasonable these plants are going to be able to make that sort of migration leap. And what this does then is mean that we have some clear expectations about what types of novel interactions might actually emerge in the future, because the plants, have clear migration tracks, and it's not unreasonable that they would be able to achieve those migration destinies. In addition, we can overcome the experimental tractability hurdle a bit because the alpine plant communities are amazingly transplantable. So we can take large chunks of alpine meadow and we can, through the use of a helicopter, transplant them to different elevations along the mountainside. And what this does is allow us to take a focal alpine plant individual, transplant that individual into any competitive and climate environment that we like along this alpine slope. And this allows us then to basically explore what will happen under different kinds of competitor or climate changes that we might forecast. Okay, so our first question is, how do these novel competitors shape species responses to climate change? And to address this question, we thought very carefully about what are the actual scenarios of competitor change that we wanted to understand. So this slide's a bit busy, but I'll walk you through it. What this does ultimately is lay out 4 different scenarios of climate and competitor change that sort of bracket the extremes of what you might think are reasonably going to happen over the next 100 years, and are ultimately the experimental scenarios that we created. So I want you to focus on the fate of this green plant here. This is our focal species, and envision that it's currently found at around 2,000 meters, the field site that I showed you already. And it currently faces a climate that's depicted in yellow. It faces a certain set of competitors, also shown in yellow. But if you move 600 meters upslope to a 3-degree cooler climate, you get a different set of competitors. If you move 3 degrees warmer in climate down to 1,400 meters, you will get a different set of low alpine, subalpine competitors. Now, this focal plant, if the climate warms, will of course face a novel warmer climate for it. But what we wanted to ask was, what will be its competitive environment? And in one scenario, if this focal plant fails to advance its range, or you look at the trailing edge of its range, it might face its current competitors, shown in yellow. If they too also persist in place, it's just a warmer climate now. But in another scenario, another extreme scenario, the plants from lower elevation migrate in and replace that focal plant's current competitors. And it's now facing these novel low elevation competitors under the warmer climate of the future. And the key question we want to ask is, does it matter for the fate of this focal plant whether it faces its current competitors or these low elevation competitors under climate change? We have another set of scenarios that emerge if the focal plant migrates upslope, or we're looking at the leading edge of its range as it expands. If it's the only plant migrating upslope, or it's the first colonizer, it will face a novel high alpine community. But if all of its current competitors come along with it, it will then face its current competitors, actually under the current climate, just in a different location. But again, we can ask, does it matter for the fate of this focal plant in its expansion, whether it faces its current competitors or these novel high alpine competitors? So as you can imagine, what we did experimentally was create these 4 different scenarios today. And what this basically amounted to was transplanting individuals of our focal species into the alternative competitive and climate environments that would emerge under these 4 different migration scenarios. So to create the scenario where the focal plant fails to migrate and it's facing its current competitors, then of course, what we do is we take individuals of the focal plant and plant it into plots that you can see here that are composed of its current competitors. But because this interaction occurs under the warmer climates of the future, we take the entire system and transplant it down to a warmer climate that's currently found at 1,400 meters in the Alps. So we give it 3 degrees of temperature increase. And then we repeat this for all the different scenarios. You could trust that we did this correctly. But for scenario 3, for example, where the focal plant is the lone migrant into high elevation systems, because it itself tracks climate, we plant individuals of it into these novel high alpine communities and transplant them to the current climate of the focal species, which is actually what you're seeing in this current slide here. And that's, of course, because this interaction would emerge by virtue of this plant having tracked climate to this high alpine site. So it's actually currently existing. It would be existing in its current climate in the future. Okay, so let me first show you the results that we have in the case where the focal plant is not expanding its range, or at least it's looking at the trailing edge of its range. So it's experiencing a warmer climate, and it might face one of these 2 different competitive environments. So remember, it could face either its current competitors if they persist in place. It can also face these low elevation competitors that would migrate in by virtue of them having tracked climate. And what I'm showing you now are the results for 4 different focal species, okay? We chose 4 different species to plant into these plots. This is, if you're curious, this is Anthillis. We have here a Plantago, a Pulsatilla, and a Scabiosa. And these plants all have ranges that center around 2,000 meters. They don't go up to 1,400. They don't go up to 2,600, nor do they go down to 1,400. And thus, these competitor environments are ones that they don't actually see in their range along the Calanda where we work in the Swiss Alps. And I'm showing you their survival after 2 years of transplantation. And what you can see is that indeed, these 4 different plants tend to survive more poorly when they're surrounded by these low elevation competitors, where the results are shown in orange, as compared to when they're surrounded by their current competitors in the warmer climates. So 3 of the 4 species show significant reductions in their survival. The other key demographic rate is probability of flowering. And again, these species also show 3 of 4 show reductions in their probability of flowering when they're growing with these novel low elevation competitors as compared to their current competitors. So what we have here is indeed, some of the first empirical validation of the idea that these novel competitive interactions can play important roles in shaping species fates under climate change. On the one hand, though, the result is in some ways quite predictable, though, because what we're showing here is basically that competitors under a warmer climate are better competitors if they actually currently live under those warmer climates than if they're currently living under cooler climates. But this was not, in fact, the result that we saw when we moved to the other 2 scenarios that I'm going to show you next. These are scenarios where the focal plant is migrating upslope and faces either this novel high elevation community or its current competitors, which migrate along with it. That's the results I'm showing you in these graphs here. Again, we have survival after 2 years. And you see that the survival is kind of the same, regardless of whether they're facing these novel high alpine communities, which have not had a history of competing or growing under these warmer conditions, or their current competitors, which actually currently live at the climates of the future. We saw a similar result if we look at the probability of flowering. There's one species here which does seem to respond favorably to these high alpine competitors, but in fact, we get the exact same result if we put them on alpine soils, high alpine soils. So we don't believe this result is caused by the competitors themselves. So it seems to be driven by the soils. I'll explain to you more about this afterwards if you're curious. But if we're going to summarize the results then from these first 2 data slides, it's that we find that the competitor shift is quite significant in the scenarios where the focal plants fail to advance their range. So growing with this low elevation competitor is very different than growing with the current competitors. But when they're at the leading edge of their range, growing with these novel high alpine competitors is not all that different than growing with the current sets of competitors these plants face. And the obvious question is why? So we hypothesized first that there might be sort of more compositional similarity between these 2 communities than between these 2 communities, but that wasn't the case. When you ordinate these systems and you calculate these compositional differences, they're equally distinct in compositional space. But even if the names of the species are sort of equally distinct, that doesn't mean the traits of the species are equally distinct. It might be that the plants are very similar in their sort of competitive traits if you compare these blue systems to the yellow systems, but very different in their competitive traits if you compare the yellow systems to the orange systems. So to evaluate this, we went back to the field and we measured 5 different plant functional traits on about 60 different individuals in the Alps in their natural environment. And then we located the different community types in this trait space. And what you can see here is the location of 10 replicate communities from 3 different elevations shown by the different colors here. So cold, warmer, and warmest. And I'm showing you the location of these communities in a trait space, showing you along the first 2 principal component axes. And the traits were leaf size, leaf mass, plant height, the area of the leaf per unit mass, as well as leaf dry matter content. And what you can see is that as hypothesized, if you look on this first principal component axis, which explains the vast majority of the variance amongst these communities in their community weighted trait means, you can see that in fact, the mid elevation communities were more similar to the high alpine communities than they were to the low elevation communities. And we believe this trait difference and the larger trait difference that you see when going from here to here, then from here to here, is responsible for the larger competitive differences that we saw when these focal plants were facing these different competitive communities. Okay, so to summarize this first portion of the presentation, I've argued that these novel competitors shape alpine plant species responses to climate change in scenarios where the focal species fail to migrate, but not when they advance their range, or at least It matters if you look at the trailing edge of the range, but not so much if you look at the leading edge of the range. Now, I don't believe this is sort of generally true. I believe the reason why we see this in our result, in our system, is because in the scenarios where the focal plants fail to migrate, we're seeing large trait changes with the novel competitors. Whereas when they advance their range, they're not facing sort of functionally very different species at this high alpine system. And that's probably the more general conclusion. Now, this is of course means that this of course is sort of lays sort of the groundwork for sort of the next portions of our presentation, where we begin to ask, can we predict which species will engage in these novel competitive interactions, and can we forecast the outcome of the interactions in my second and third question here. Okay. So what I want to do now is turn to the second question about predicting the migration velocities of species. Can we predict what species will be engaged in these novel interactions in the future? Answering that question requires us to be able to predict how fast these taxa will migrate under climate change. Now, this challenge is simplified to some extent by the fact that there's a large body of ecological theory that kind of tells us how populations expand. And what it tells us is that if you're looking at a population that's expanding in this direction, towards the right on my screen at least, spreading populations tend to be what's called pulled by rare individuals at the front of the migration. So it's these leading edge individuals which disproportionately contribute to the expansion of the population. There's many, many more seeds produced behind the invasion front, but the disadvantage of being farther from unoccupied sites is simply so overwhelming that their contribution is effectively zero. And what this means is that you can simplify the migration process and predict the migration process if you know two things. You need to know the dispersal capacity of the population, so how far individuals disperse away from the mother plants, as well as the fecundity of those plants when they're at low density. Again, performance at high density is irrelevant, so we don't need to know how well they can handle density dependence. And what you get then is a prediction of an expansion velocity, which is constant. So the population moves at, on average, the same rate through time. Now, this is very useful, and this information has in fact caused ecologists that are trying to forecast the future ranges of species to constrain, for example, the climate-driven range shifts to what's maybe possible given knowledge of species dispersal and migration capacity. And that's a very important development. But what I want to do today is address a related question, which is, should we even expect these migration velocities to be constant? Or in other words, what's the role of a factor such as evolutionary change in changing these spread velocities over time? And the reason why this is an important question is we expect selection on individuals at the front of the invasion for greater dispersal, because greater dispersal allows these individuals to essentially found the entire rest of the expansion, which is of course quite advantageous in a sort of spatial sense of selection. But we also expect these individuals to experience strong selection for fecundity at low density, even if it comes at the expense of tolerating competition when at high density, because that will again lead to the great number of individuals found in subsequent generations as you look at the fronts of these invasions. So as a byproduct of this selection on dispersal and fecundity at low density, we expect evolution to lead to essentially a faster and faster spreading population. So individuals, if the population front is experiencing essentially greater dispersal and greater fecundity at low density on account of evolutionary changes, then we expect that front to move faster and faster in successive steps. And this, of course, is bad news if you want to forecast the migration of taxa with currently measured vital rates, or even more challenging, forecast which taxa will come together and face one another in a competitive environment in the future, because it suggests that in fact you might need to account for evolutionary changes if you want to understand the migration velocity. Now, everything I've described thus far is sort of classic ecological theory coming from landscapes that are uniformly favorable. But we know that sort of the migration that we're interested in is often one that occurs over landscapes that are fragmented by anthropogenic features, such as roads or agricultural practices, but also natural features of the environment, such as ecotones or rivers and so on. And what this means is that we want to make sure that our rules for how populations spread are applying to these fragmented landscapes, which are, of course, almost the norm in the landscapes that we want to understand climate change migration in. And we know, for example, that from ecological theory, that the patchiness of the landscape has a big effect on the evolution of dispersal. And our own work has shown this can also affect this can also introduce a role for the tolerance of competition, because in highly fragmented systems, populations often build up to high densities before they're able to land a seed in the next suitable patch. And this can mean that tolerating competition becomes an important trait for the ability to disperse as a population to the next patch. Okay, so the two questions we had in this second portion of the presentation are, does evolution on ecological timescales and expanding populations increase the spread velocity? And secondly, does landscape fragmentation influence the potential role of evolution? So ideally, we would examine these questions with sort of naturally range expanding populations that you can see in the foreground of the slide. But the problem is basically that spread, especially in plants, occurs over such large spatial scales and such long time scales that it's very hard to get sort of a mechanistic or process level understanding of what's happening in an expanding population in these systems. So for this reason, we've been developing a laboratory system to study spread. This is work that was led by Jennifer Williams, who's now a professor at the University of British Columbia. And here we worked with Arabidopsis thaliana, the common sort of plant lab rat or genetic model system, to study spread under controlled conditions. And Arabidopsis is ideal for many reasons. The most obvious is that it's very short generation times. So you can get 3 to 4 generations per year in an experiment that we will conduct. The plants are also small, meaning that they don't disperse terribly far. So you can study spread at the scale of a greenhouse, which is not overwhelming. And then finally, because this plant's sort of a genetic model system, there's been a variety of recombinant inbred lines that have been developed for these species. These recombinant inbred lines are very useful because we can essentially buy them, we can propagate them, and this allows us to examine populations that have genetic variation, which of course is required for there to be any evolutionary change over the course of the spread process. So the basic protocol for our experiments involves setting up an array of pots, a linear array of pots that you can see photographed here. And we sow the plants into the leftmost pot of the array. The different colors are meant to indicate different genotypes. We begin our experiments with 14 different recombinant inbred lines. And then we let the seeds germinate, they make plants. The plants make seeds or make flowers, which then make seeds. And then we help the seeds disperse by imposing an artificial rain event, which essentially is spraying them from above. And then after a generation, we might find a pattern of seed dispersal, which looks something like this, where we see that the populations have expanded down the runway. And in addition, there's also been some change in the frequency and spatial sorting of the genotypes. The blue ones, for example, have made it towards the front. The red ones have stayed towards the back. Some have, like the green one, have disappeared altogether. And what this illustrates then is that not only is there ecological change over generation, but there's also going to be a change in the genotype frequencies and their spatial patterning. And what we want to then evaluate is do those genotypic changes then affect how fast the populations can expand in the subsequent generations. So to evaluate the role of these evolutionary changes or clonal sorting, if you want to be a stickler, how will these genotypic changes affect the spread velocity? We need some sort of treatment where we prevent these genotypic sortings as well as changes in frequencies from occurring. So we have a non-evolving treatment. It's not entirely non-evolving, I'm aware of that, but it's a shorthand for the treatment. And what we do with this non-evolving treatment is we essentially have a no evolution control where we let the plants go through the exact same process that I showed already. They make seeds, those seeds disperse. But what we do is we then let those seeds germinate, but then we kill all those individuals and we replace them with individuals that have been randomly drawn from the initial seed pool that began the experiment. So it'll turn this type of system to one that looks like this. And what this does is perfectly retain the prior generation's ecological dynamics, but it removes any change in genotype frequency or spatial sorting of those genotypes. Okay? And then the comparison of the spread velocity in these two treatments allows us to evaluate the role that these genetic changes play, or these genotypic changes play in the spread velocity. And then finally, to examine the role of this evolutionary process as a function of the landscape patchiness, we can of course remove pots from the array. And we do a variety of pot removals. I'll show you those results today from ones that have pots that are so far apart that they're about 12 times the mean dispersal distance of an Arabidopsis seed in this study. Okay, so the first result I can show you is that indeed these populations do spread sort of along the lines of what ecological theory would tell you they should spread. So along the x-axis here is the distance they've moved in the experimental runways. The y-axis is the number of seeds that they have in each one of these pots. And this is an individual replicate, okay? So the replicate began in this pink pot here at 0.0. It made some seeds that led to the red distribution of seeds in the next generation, then the orange, then the yellow, then the green, then the blue, and so on. And you can see the populations have indeed moved down the runway. And they moved down the runway about 1.5 meters over the course of 2 years, which is not very impressive, I recognize, but we don't need this to be impressive. We see them to move. And then we can ask, does this spread process depend on whether or not the populations have the chance to evolve or not over the course of this eco-evolutionary process? And here's the results. So I'm showing you results for the continuously favorable landscapes and the most patchy landscapes, landscapes where the gaps between suitable patches were 12 times the mean dispersal distance. On the x-axis is the generation of the experiment. So we ran it for about 6 generations. And on the y-axis is the furthest dispersed seed. So how far did the population get in any one of these runways? And each one of these green lines shows you an individual replicate's dynamics that's evolving. The gray dashed lines show you a non-evolving replicate. And then the dark green and the dark black dashed lines show you the mean for these two treatments. Okay, so what we found in the continuously favorable landscapes is that giving the populations a chance to evolve as they spread did have some effect on their ability to expand. So we see that the populations are moving about 9–10% faster when they were given the chance to evolve versus when not. But this difference is not significant. The p is only around 0.06. But also, I think it's worth considering that all of the sources that there's lots of sources of uncertainty in the projection of a spread velocity for a population in nature. And knowing that, in fact, these populations are spreading maybe too fast or too slow relative to our predictions because we fail to account for evolution is not going to change the way we approach this problem, because this effect is simply just not all that strong. But if you move now towards the patchy landscapes, we now see that the effects of evolutionary change are very strong on the expansion velocity. We're seeing that the populations moved about 3 times as far when given the chance to evolve as compared to when they didn't have the chance to evolve in these patchy landscapes. And moreover, this happens fast. This is 6 generations of change. This is on the time scale that you'd want to think about evolution affecting the velocity of a climate change migrant. So the obvious question that follows from this result is, why do we see these large effects of evolutionary change in the patchy landscapes on the spread, but not so much in the continuously favorable landscapes? Well, we have the ability to actually evaluate how the traits have changed in these populations, because if you recall, these are clones. And what I forgot to mention, these clones basically don't outcross. They almost entirely self, these individuals. These plants are almost obligate selfers in these experiments. And what this means is that we can then snip genotype the individuals that are at the leading edge of the invasions or the expansion fronts, and we can assign them to one of the different recombinant inbred lines that began the experiments. There's 14 recombinant inbred lines. You can imagine that these 10 leading individuals fall into 3 of them, with recombinant inbred line A being most frequent. And then we can also grow these recombinant inbred lines in common garden settings and measure a variety of spread relevant traits, such as their height, their competitive ability, their dispersal ability. And then we can calculate a mean trait value for each of these populations, basically weighting the trait values by these different frequencies of the recombinant inbred lines that you find in the population. And this allows you to reconstruct the trait evolution over the course of the experiments for all the recombinant lines in all of our replicants. So this is the results of this effort. So along the, I'm showing you again results for continuous landscapes and patchy landscapes. And I'm showing you the locations of each of the evolving replicates, shown by the black points, in a trait space that's defined by competitivity, dispersal, and plant height. And the orange stars in these graphs show you the progenitor populations, their locations in that trait space. And what you can see is that these populations did indeed diverge from the progenitor populations over the course of the experiments. Now, our first hypothesis for why we saw faster spread when given the chance to evolve in the patchy landscapes, at least a bigger difference here than we did in the continuous landscapes, was maybe we just saw more rapid evolutionary changes in the patchy landscapes. So to test this, we simply calculated the difference between each one of these evolving replicates and the founding population, and asked, was there more trait change over the course of the experiments? And when we calculate those Euclidean distances, what you can see is that the lines are kind of the same in the two treatments. They evolved in both treatments, just fine. But what you'll notice, and is more obvious, is that there's a different evolutionary trajectory in the two treatments. So in the continuous landscapes, they tend to essentially evolve all over the map. You'll probably see the effects of essentially of allele surfing, which is a case where basically you have genetic drift on steroids in a spreading population. But in the patchy landscapes, we see rather consistent evolutionary changes. And the populations essentially move further to the back of this graph and further to the right of this graph, meaning that the populations evolved to be taller. And on account of that, also were able to disperse their seeds farther. So we believe that this is the reason why we see the consistent evolution of, we see the consistent faster spread of evolving populations in the patchy landscapes and less so in the continuous landscapes. The final point I want to make is you get a similar message if you don't look at the trait change, but just the genotypic changes. So here you can see the actual frequencies of the different genotypes. Here's the founding population composed of 14. You'll see there's large genotypic changes in all of the treatments, in both treatments. It's just that in the continuous landscapes, these genotypic changes go all over the map, whereas in the patchy landscapes, they're more consistent towards one or two of the genotypes dominating. Okay. So to summarize the second portion of the presentation, I've argued that evolution on ecological timescales and spreading populations can increase the velocity of spread, especially in fragmented systems. And what this means is that estimates of spread velocity based on current demography may underestimate the eventual expansion rate of climate change migrants. So what I want to do now is turn to the final portion of the presentation and say that if you could ignore the second message, and it was indeed possible to forecast which species will come together under climate change and compete in the future, could we predict what species will win based on knowledge of their functional traits? And this is important because the climate change migrations are going to present plants with countless novel interactions. So the red one, for example, here faces the orange and the green and the blue, but even more pronouncedly, if you look at any one of these locations along the alpine gradient, you find a whole suite of competitors. And it's just not practical to be able to go in and do the kind of experiments that I showed you at the beginning of the talk on a species by species basis. Thus, we need some efficient proxy for the kinds of results that we would find in those experiments. And plant ecologists have classically turned to functional traits to provide those efficient proxies. These can be whole plant traits like plant height or seed size, or leaf traits such as leaf nitrogen or leaf mass per unit area, or leaf area per unit mass. And the reason why these traits are thought to provide important proxies for competitive interactions is the classic prediction that competition between species with different sets of traits should be weak. So if you locate your plants along a leaf trait axis and a plant size axis, plants that are far apart from each other in that trait space should have weak competition, whereas plants that are close together in that trait space are thought to have intense competition. And the reason why we make this prediction is we believe these plant functional traits are telling us about their niche differences. Differences between species that cause them to limit themselves, more than they limit other species. So for example, these two plants differ in their rooting depth, and we would argue that if in fact plants truly can partition soil resources with depth, the deep-rooted plant will exert a bigger effect on other deep-rooted individuals than it will on individuals of the shallow-rooted competitor. And the reason why this is thought to lead to weakened competition is because at the population level, if any one of these species becomes increasingly common and starting to really exert strong competition on the other species at the population level, this strong intraspecific limitation relative to interspecific limitation will keep those populations and their competitive interactions in check. So if in fact this was the way in which all functional traits worked in nature, then in fact our job as empiricists would be very straightforward. We simply need to quantify which of these functional traits are highly predictive of this weakened competition. But the problem is that sort of contemporary coexistence theory teaches us that this in fact is not so simple. And the issue is that functional traits might in some cases predict sort of weakened competition, but in other cases predict the exact opposite. They might actually predict the competitive imbalances between species. They might predict what are often called the fitness differences in the coexistence literature. These are differences between species that simply favor one competitor over others. Maybe this deeper rooting depth is simply a superior strategy, and is going to allow this plant to win in competition with the shallow depth. Another example might be differences between species and their R star, their ability to draw down a common limiting resource. We don't expect these differences to lead to weakened competition. We expect these differences to lead to the elimination of the species that is more poorly able to draw down that limiting resource. So functional traits might predict these types of fitness differences as well. Thus, the main message is that ecological theory does not give us any clear guidance as to whether or not trait differences should lead to weakened competition or, in fact, large competitive imbalances. Thus, the job is for empiricists to go to nature and ask, to what extent do functional trait differences predict niche differences versus fitness differences between competitors? Will they predict the differences that weaken competition, or will they predict the differences that accentuate competitive imbalances? And we should know this if we're going to then forecast the outcome of competitive interaction between two novel competitors that come together in the future. So we've been addressing this question in annual plant communities in California. And we've chosen these annual plant communities because the plants basically have relatively simple life cycles that lend themselves to mathematical model descriptions. And this is important because only once you have a mathematical model of the composition between the species that you believe, can you actually generate a theoretically justified measure of those niche differences and those fitness differences that I showed you pictorially in the slide before. So what we do in this annual plant community here in California, and this is work that was led by two former postdocs that are now professors at the University of Cadiz, Oscar Godoy, and UCLA, Nathan Kraft, is we grow these 18 annual plant species in pairwise competition with each other. So we grow them in a way where each species faces a density gradient of competitors that allows us to actually quantify the per capita suppression that all these individuals have on one another. I'm happy to explain how we do this more later on, but you can trust that we can get these interaction coefficients from our experiments. This is classic plant ecology. We can also get all of the demography of these species as well. So the growth rates or their fecundities with no competitors, their germination rates, their survival rates in the soil, and so on. And then with our mathematical model specified, we can then quantify the theoretically justified measure of the niche difference between the competitors. How much weaker is the competition between the species than it is within species, what you see from this metric here. So this is actually the ratio of the intraspecific limitations to the interspecific limitations, okay? And this indeed will tell you whether or not species get advantages when rare, disadvantages when common, which will ultimately stabilize their coexistence and weaken the competition between species. We can also quantify the functional traits the plants have, so the rooting depth, for example. And what this allows us to do is for a given pair, make a point in the graph relating the niche difference between the competitors and their trait difference. And then because we do this for all of these different plant competitors, we can look across species and get lots of points. And then we can ask across the competitors, how well do the trait differences that we measure between a species predict the niche difference? We can do the exact same thing for the fitness difference. I'm not going to show you the expression for it, but you can imagine using a different combination of demography and competitive interactions to quantify the competitive imbalance between the species, and we can do that. And we can again relate that to the trait difference between the competitors. Okay, so here's the results. What I'm showing you first is the correlation between the trait differences between different competitive pairs and the niche differences between different competitive pairs. And the correlation can naturally range from -1 to +1. That's how you read this graph. And the intersection of the red line with the axis for a given trait tells you how well the trait difference predicts the niche difference between the competitors. And if it falls within the gray shaded region, that trait difference is a non-significant predictor of the stabilizing niche difference. And what you can see is that for all of these different plant functional traits, this is the suite that we measured on our species. Here we have a measure of water use efficiency, whole plant water use efficiency. We have area of leaf per unit mass. We have length of root per unit mass of roots. The rest are self-explanatory. The key point is that none of these are significantly predicting by themselves the strength of the stabilizing niche difference. And this is important because not only does this tell us that if two plants come together in the future and are very different, we should expect weak competition. But there's a whole body of sort of trait-based sort of community ecology, which attempts to try and use the trait differences between competitors to actually back infer the strength of competition. And what this work is telling us is that, in fact, if you try to relate trait differences to a theoretically justified measure of competitive dynamics, you're not actually finding a relationship along the lines of what this sort of trait-based community work would suggest. Gets even worse. Here we have the correlations between the trait differences and the competitive fitness differences. And what you can see is that these, now we actually are picking up predictions. So in fact, we're finding that not only do these trait differences fail to predict the weakened competition, as sort of classic community ecology would lead you to believe, in fact, they're predicting competitive imbalances. So plants with larger leaves, plants with less area per unit mass of leaf, plants with lower leaf dry matter content, plants with higher leaf nitrogen in their leaves, plants that are taller, plants that root deeply, plants with less root length per unit mass, and plants with a later phenology all tend to win against species that have the opposing trait values. So in fact, what we predict here is that in fact, the trait differences are predicting the competitive imbalances between species, not the weakened competition that you would predict from a stabilizing niche difference. So the trait differences are useful, but not in the way in which we classically pose. So to summarize this final result, it's that the strength of the fitness differences rises much more steeply with the trait difference between the competitors than does the strength of the niche differences. And what this says is that on balance, these trait differences are going to generate competitive asymmetries. Now, this doesn't mean that in fact, niche differences are not underlined by trait differences. We know they are, they have to be. But it suggests they might not be underlined by the commonly measured trait differences that plant ecologists commonly use. It might instead be the trait differences that are more challenging to measure, that maybe relate more specifically to resource uptake, for example, or more detailed information about the interaction between the competitors. And the final point I want to make is that, in fact, this result, that trait differences generate competitive asymmetries, is one that I used early on in the presentation. If you recall, I argued that the reason why our alpine plants were so sensitive to a shift in competitive identity when you move towards these low alpine systems, as compared to a shift towards these high alpine systems, was because only in these low alpine systems did we see large trait changes. I was essentially arguing that these large trait changes were associated with competitive imbalances. Okay, so to conclude from the whole presentation, I've argued these novel competitors may strongly determine the fate of species under climate change. We then argued that predictions of population spread based on current demography may underestimate the eventual velocity. And this is a real challenge for trying to forecast the emergence of these novel competitive interactions going forward. And then finally, I argued that even if we can predict which of these novel competitive interactions are indeed going to emerge, functional traits might actually be very useful for forecasting the outcome of the interaction, but not in the way in which we classically think. It's not so much that they're going to tell you which pairs are going to have weak competition. It's more they might actually tell you which pairs are going to be the ones that will see sort of large competitive imbalances. And then we need to do further work to determine what in fact is the optimal trait value under those future climates. And then the final, more holistic message is that there really is, for me, a connection between these fundamental curiosity motivations for ecology and the ecology that's solving global environmental challenges, in the sense that we had been working on questions about trait-based predictors of competitive dynamics, as well as the controls over spreading populations for years before we actually began working on these alpine plant community responses to climate change. But only once we got into this more applied question about plant community responses to climate change did we realize that the major knowledge gaps were the ones for which our more fundamental science actually held the relevant answers. So with that, I'd like to finish up, acknowledge my collaborators and funding sources, and be happy to take any questions. So thank you.